Okay, thanks, Gareth. Afternoon, everyone. Really good to see so many people up on the call from various places. And it's a pleasure for me to introduce Mark Russell, who's going to be today's speaker. I've known Mark for quite a long time. He first joined our lab, I think, as a technician 20 odd years ago, graduated through all the intermediate phases, PhD student, postdoc, and is now forging his own furrow as a lecturer in Exeter. And most of that time he's been interested in signaling mechanisms in the beta cell, particularly anti-inflammatory signaling pathways and the way they intersect with the pro-inflammatory mechanisms. And I think that's the kind of territory that Mark's going to explore with us today. So it's great to see you, Mark, and we'll hand over.
Lovely. Thank you very much, Noel. I will just now share my screen. Okay, so hopefully that is visible for everyone. Can I just clarify it's how long I've got to give this? Is there another presentation?
There is no other presentation, so yeah, you've got as long as you want, really.
Don't say that, I'll be here all day.
We need to go at 5, Mark.
Right. So yeah, thank you very much for that introduction, Noel. Yeah, 2005 is I think when I started in Plymouth, so it's been quite some time and quite a ride over the years with some fabulous colleagues along the way. So my interests, as Noel has suggested, relate to many of the mechanisms which control beta cell survival, and I've spent a long time looking at both anti-inflammatory and pro-inflammatory mechanisms, particularly around cytokines that control that. And one of the pathways that's been central to much of my research has been the JAK-STAT signaling pathway, which I'm going to be telling you a little bit more about later on. But I think what I wanted to focus this talk on was probably one of the most exciting advancements in this particular area in recent times, given that there has been a paper that suggests that this particular pathway may be able to be used as a potential treatment for type 1 diabetes. So I want to look at the sort of background evidence for this and then move forward to looking at the data that they identified in that paper. So for I think this audience probably does not need reminding that type 1 diabetes is an autoimmune disease in which the pancreatic beta cells are selectively destroyed by influent immune cells. And we know there are a number of hallmark features which are important within type 1 diabetes, a couple of which I've alluded to already: the loss of functional beta cell mass, the reduction, the increase in infiltrating immune cells at the islet site. But there are also other hallmark features that have been identified, and some of which have been identified quite some time ago. And one of these is the elevated expression of what we call the human leukocyte antigen class 1, or HLA1, on pancreatic beta cells. And I'll be referring to this as the hyperexpression of HLA1. And this was first discovered back in the 80s by people like Franco Batazzo and Alan Fowlis and others, looking at pancreatic sections and staining these for HLA1. But in more recent times, we've explored these more carefully. So what is HLA1? So this is an antigen-presenting molecule or antigen-presenting complex expressed on all nucleated cells. So its job in the cell is essentially to present peptides from within the cell, which represent the repertoire of all the peptides expressed within the cell. This will include those endogenous peptides, but also if there are any non-cell peptides present within the cell, these will be presented on the surface to CD8 positive T cells. And if there is a CD8 positive T cell that is able to recognize that peptide attached to the HLA1 molecule, for example, if it is a virus, then you can get selective killing of that peptide. So it's a very useful mechanism for clearing, for example, infected cells. So in the context of type 1 diabetes, what does HLA1 actually look like? Well, this was some work done from the team in a study led by Sarah, I think in 2016, where we looked at HLA1 within the pancreas of a whole range of individuals with type 1 diabetes from an array of different cohorts using a whole array of different mechanisms. And so this was using some immunoperoxidase staining, and we're staining here for HLA1 in the brown. And this is a sample from an individual without diabetes, and you can see in the center there is an islet, and that's staining, relatively weakly for HLA1. So when we then looked in the pancreas of people with type 1 diabetes, we saw a subtle difference. I think you'll appreciate it's a huge elevation in the expression of this HLA1 molecule. When we did this using different approaches, this time an immunofluorescence approach, labeling HLA1 in the green, we can see this elevated expression of HLA1 again. And using different fixation approaches, the previous two are FFPE, but this one now is using in frozen sections. We can see again in the red here this elevated level of elevated expression of HLA1. We were able to see this across multiple cohorts, from data from multiple labs, using different antibodies, and also using RNA-seq analysis of isolated laser captured islets from these sections. So in almost every mechanism that we were able to explore this, we were able to see this HLA1 validating the earlier studies that I mentioned. So does this even matter? What's the point of all this? Well, in the context of type 1 diabetes, this is quite important, because type 1 diabetes, as we know, is an autoimmune disease, and the immune system is targeting, in this case, beta cell proteins. Now, as I've just mentioned, for the function of HLA1, it's going to be presenting these beta cell proteins on the surface, and some of those are going to be autoreactive. So essentially what's going to be happening here is that the beta cells are going to be hyper visible to the immune system due to this elevated level of HLA1, and ultimately this is likely to increase the rate of beta cell death. So the thought process here could be that if we could somehow block this from occurring, then this might provide a therapeutic mechanism to delay or slow the disease. But to be able to do that, we need to understand how HLA1 how the mechanism of HLA1 upregulation. And this is something that we've been exploring in Exeter, and others have elsewhere over the past few years. So one of the, if you look in the literature, one of the mechanisms by which HLA1 gene expression can be regulated is through interferons. Interferons are a class of cytokine which are released from a variety of different sources. So some of these are released in response to viruses. These are things like type 1 interferons, the likes of interferon alpha, and type 3 interferons such as interferon lambda. So they are antiviral cytokines released by virally infected cells to let other cells around them know that there is an infection occurring. On the other side, we have these type 2 interferons, which are, for example, interferon gamma, and this is typically released from immune cells and acts as a pro-inflammatory cytokine. Now, in the context of type 1 diabetes, both of these events are likely to occur. Fantastic work that's been done from Sarah and others has shown that viruses have an important role in potentially triggering type 1 diabetes, as beta cells are a target of certain enteroviruses, particularly Coxsackie B4 viruses. And then there is then an improper response to those viruses, which then perpetuates the disease process. But we also know we have immune cells that infiltrate the beta cells as well. And these are going to be releasing an array of different cytokines, including the likes of interferon gamma. So the sum of this is that beta cells are likely to be exposed to an array of different interferons during the process of type 1 diabetes. Now, there's some key components here that you need to understand, and I'll just explore these components. So we have our receptors, our cognate receptors, and attached to these receptors, we have these JAK proteins or Janus kinases. There are 4 of these, but only 3 of these are linked to these receptor tails. Upon binding of a ligand to the receptor, we get activation of these Janus kinases, and these then lead to downstream signal transduction. So downstream of these JAKs are another group of proteins called STATs. So these are the cleverly named signal transducer and activators of transcription. That is exactly what they do. They're transcription factors, and essentially these are activated by JAKs by phosphorylation. And upon phosphorylation, they dissociate from the receptor, form dimers in the cytosol to induce changes in gene transcription, as shown here. And what we think is going on is that HLA1 and the genes that encode HLA1 are some of the genes that are downstream of these STAT proteins. So, is there any evidence that STAT1? Oh, I should say, what I wanted to say is that what you'll see if you're being particularly vigilant is that STAT1 forms part of both of these dimers. So STAT1 may be quite important in both types of these signal transduction cascades. So, given that, is there any evidence that from within the tissue that there is any sort of link between STAT1 and HLA1? So we asked that question, or similar questions, that when we were studying the HLA1 hyperexpression in that 2016 study back into a few years ago. And here we can see some staining where we've stained the same islet for HLA1, and this is in the red, and STAT1 in the green. This is from an individual without diabetes, and you can see there's very little evidence of STAT1 or HLA1 staining in those in the islet. And you can see this is an insulin-containing islet because we've got light blue over here, which is your insulin. So we looked in islets from people with diabetes, type 1 diabetes, but these are the insulin-deficient islets. So this is an islet without any insulin. Saw a very similar pattern. But when we looked in the islets from people with type 1 diabetes, which contained insulin, we see this elevated hyperexpression of HLA1, and we also see this elevated we see an elevation of STAT1 expression as well. And we saw this consistently across all the sections that we were studying. And interestingly, when we looked to correlate the expression of these two molecules, what we saw was that the mean fluorescence intensity of STAT1 from an islet and the mean fluorescence intensity of HLA1 in a given islet correlated very nicely. So the more STAT1 you had, the more HLA1 you had. And we saw very similar patterns across a multitude of different donors. Okay, so we wanted to explore the role of interferon then in driving STAT1 and HLA1 signaling within pancreatic beta cells. So we used the gold standard human beta cell line, the endo-C beta H1 cells, and we just exposed these to different species of interferon, either for a short time of 30 minutes or a longer time of 24 hours. And what we found, as expected, is that phosphorylation of STAT1 is dramatically increased in response to both of these types of interferons, interferon alpha and gamma. And this, after 24 hours, reduced somewhat compared to what we saw at the early time point. This mirrors what other people had seen in other cell types. What was really interesting, though, is when we looked at the levels of total STAT1, the expression of total STAT1 was markedly elevated after 24 hours of treatment with interferon alpha or interferon gamma, which is remarkably similar to what we were seeing within the pancreatic tissue. So we wanted to understand the dynamics of this expression, and we performed an experiment to see whether this was maintained over a period of time. So this is a slightly more complex experiment here, where we've taken our endo-C beta H1 cells, we've grown them, we've treated them in the presence of interferon gamma for 24 hours. So at this 0 time point, they've been in there for 24 hours. You can see this elevation in total STAT1. But then we wanted to understand, well, if we remove that interferon, does the STAT1 remain elevated, or does it reduce back down to baseline? And to our surprise, we saw that STAT1 levels were consistently elevated up to 144 hours after interferon gamma withdrawal. So this sort of suggests that this the STAT1 expression is persistent over time. So we then will look downstream of that to see whether interferon could do similar things to HLA1. And this was done by a postdoc in the lab, Puriya Akbari, who was looking at the expression of HLA1 in response to a variety of species of interferon. Apologies that this has got PIC on there, ignore the PIC. The control is in the bottom left, and you can see that the surface expression of MHC1 was elevated with each of these different classes of interferons, which was very similar to what we saw with STAT1, showing that HLA1 is downstream of this interferon signaling in beta cells. So again, in endo-C beta H1 cells, work from one of our PhD students, Mohammed Beatty, who did some fantastic work in the lab. And this was probably his key bit of data that was really, really quite exciting. Sadly, he never actually finished his PhD, and he passed away before he was able to complete it. But yeah, we're very grateful for having Mohammed in the lab and for him generating such wonderful data. And what he did was a very similar experiment to what we'd seen with the STAT1, but this time looking at HLA1. And what he was able to show was, again, after 24 hours of treatment with interferon gamma, we saw this elevation of HLA1. And then after withdrawal of interferon gamma, the HLA1 remained upregulated for up to 144 hours. So again, we see this persistent upregulation of HLA1, persistent upregulation of STAT1, which is very reminiscent of what we see in the tissue. The question is now, do we also see, this is all from cell lines, do we see similar responses in islets? Well, I've taken some data here from a fabulous study from Trivedi et al. From Helen Thomas's lab, which was published in 2017. There's a lot of really nice data. I'm going to present a little bit more of it in a second, but what they showed in this study was that if mouse islets and human islets treated with interferon gamma also showed elevation in MHC class 1 expression on beta cells. So interferons are driving HLA1 expression in our cell models, and also in these islets as well. So this is very consistent with what we're seeing in the tissue. So how do we? So given this interferon signaling pathway appears to be driving HLA1 expression, what can we do about this? Are there areas that we can use to block this, perhaps? And there are a few areas that we could target. The interferon receptor itself is certainly one area, and I'm aware of I think a couple of interferon receptor blockers that are used in other contexts, which could potentially be fruitful in type 1 diabetes. We could look downstream at the STATs and use STAT inhibitors, small molecule inhibitors for STATs. Unfortunately, these are proving very, very hard to design. There are some out there for STAT3, but those have come after a lot of work. I'm not aware of any specific inhibitors available for STAT1 or STAT2, certainly not that are in clinical usage at the minute. So that leaves in the center our JAKs and whether JAK inhibitors might be the way to go. And actually, these are quite amenable to inhibition. So there are a number of JAK inhibitors which have been generated over a period of time. I think we're on about the third generation of these JAK inhibitors, and they are getting more and more specific for individual JAKs. You can see on the right there some of the JAK inhibitors that have been used in the context of type 1 diabetes research, and some of these have been starting to be used within clinical trials. So do these JAK inhibitors, are these effective in the context of diabetes? Well, let's have a look at data from the NOD mouse. I'm going back to this Trivedi study. As I said, it's a cracker of a study. I really recommend having a read of it. And they used an AstraZeneca drug 1480 in NOD mice, and they were looking at whether these JAK inhibitors could be beneficial in these NOD mice. And what they found was really quite interesting. So they looked at a whole range of things, and I'm not going to talk through everything, but they looked at HLA1 expression on the islets in these mice, and what they found was that the JAK inhibitors were able to block the elevated HLA1 expression in these mice. They were also able to reduce the immune cell infiltration in these mice. You can see in the vehicle control the dots there as the in these mice which spontaneously develop diabetes. You can see the number of immune cells is going up, but in the squares there you can see that this is inhibited with the drug. So this is really nice data as well. But the big question is, does it have an effect on diabetes development in these mice? And the answer was very much yes. So here we see this Kaplan-Meier curve. Again, here the dots are your vehicle, the squares are your inhibitor. And you can see here there was a delay in the development of diabetes in these mice, and in other data that I'm not showing, they did find that there did seem to be some effect, some ability to reverse diabetes in newly diagnosed diabetes in these mice as well. So these inhibitors appeared to be really quite effective. They went on to do, I think, a couple of other mouse studies which are now published. All of these looked with different JAK inhibitors, and all of these looked very similar in terms of their benefits. So, does this work is this going to work in humans? And the reason I wanted to present this is because of this study, which was presented it was either January this year or December last year. I can't quite remember. But this came out of the work from Helen Thomas's group and all of these mouse studies. And this was a trial that they called BANDIT, which I think stands for baricitinib in type 1 diabetes or something like that. And they were basically seeing whether this JAK1/2 inhibitor baricitinib could be beneficial for people with type 1 diabetes. So the BANDIT trial was a double-blind randomized placebo-controlled trial. Type 1 so the inclusion criteria was that type 1 diabetes needed to be diagnosed in the previous 100 days. So this needed to be soon after diagnosis. And essentially these participants were given 4 milligrams of baricitinib, which they would be delivered orally for 48 weeks. And the primary outcome was mean C-peptide levels after a mixed meal tolerance test. I think one thing to note about this trial is that the average age of these individuals, as you can see, it is 18. So this is quite old, and I think that the data that Pierre and others have generated around the two endotypes of diabetes would suggest that many of these would be in your type 2 endotype, and perhaps not that type 1 endotype, where you might have a more sort of immune a more defined immune response there, which might be which might be more responsive perhaps. But we'll have a look what they found, because I think it was still very beneficial. So this was their primary outcome, and as you can see in the placebo in the grey bar, over time we're looking here at C-peptide levels, and we can see here as time goes by, C-peptide levels are decreasing over time, and baricitinib is able to preserve those. So that is very nice, and it does that significantly. So that's very good. What about the secondary endpoints of the trial? Well, they also looked at insulin dose and found that baricitinib treated patients required less insulin. So that's another really nice outcome from this trial. HbA1c wasn't really affected. But they also found some very interesting things around glycemic control. So there was less variation in the baricitinib treated individuals in terms of their glucose variation. In terms of time in range, the baricitinib was within the defined range that they wanted to be in for longer, above range for less time, and below range for less time. So all in all, it looks very promising. So in summary from that study, the data suggests a beneficial effect of baricitinib in type 1 diabetes versus the placebo. They showed increased C-peptide, reduced insulin dose, improved glycemic controls. They also, I didn't show these data, but they also showed a reduction in CD8 positive memory T cells in the blood, and they did some small bit of functional data to look at phosphorylation of STAT3 to see whether there was actual effects on the JAK-STAT signaling pathway, and they saw a reduction in phosphorylation of STAT3. So this did look, so I think the data looked really, really promising. So what I've told you is that HLA1 is hyperexpressed in type 1 diabetes, in islets from people with type 1 diabetes, and this is likely a contributor to beta cell loss. Interferons promote the upregulation of HLA1 in beta cells, and this is via a STAT1 centric mechanism. Blockade of this pathway by JAK inhibitors can prevent this upregulation of HLA1 both in vitro and in vivo, and these JAK inhibitors appear to be beneficial in models of type 1 diabetes and also in clinical trials. So some additional thoughts which might be worth just thinking about. Are we looking at the right populations for these trials? We've touched on that. Do we need to, are these going to be more effective in younger individuals? That's perhaps. Are there better mechanisms to deliver these drugs to the islets more directly? We know that these JAKs are likely, they're expressed throughout the body, and therefore we might well see many off-target effects. So bringing these drugs, or targeting these drugs more effectively to beta cells, might allow for a better mechanism to get a better effect within beta cells, but also reduce some of those off-target effects. Question is, are we targeting the correct JAKs? These are JAK1/2 inhibitors that we're talking about. Do we want to target in on JAK1? The reason we might want to do that is that JAK1 forms part of both the interferon gamma and the interferon alpha pathway. So we might be able to, just by targeting one JAK, hit two birds with one stone. And should we be exploring other mechanisms to target this pathway? I think this might well be an important thing to do. And in fact, we're doing some work looking at novel ways to regulate STAT1 activity, which I'm sure at some point we'll present in the future. Fantastic. So that is everything I had, and just a big thank you to the IBEX team, and particularly those people mentioned there who directly contributed to this study, but there are some amazing people within IBEX who aren't mentioned there, so thank you to one and all, and the funders.
Thanks, Mark. That's really good. Really excellent talk and very clear. And I'll invite people to. Do you want to stop sharing so I can see people on? We can see everyone's on screen. Please put your hand up if you want to ask a question or make a comment. Perhaps since I'm sitting here and got my microphone unmuted, I'll kick off. Can I ask one about the non-classical HLA ones? Are they also likely to be down-regulated under these conditions? If you use the baricitinib or other JAK inhibitors,
I think what you're touching on there is, I think, a really important thing, because obviously we are going to be targeting other things within the islet, and that potentially would be these molecules would be potentially targeted, as well as perhaps some of the protective pathways as well, the likes of the IL-13 signaling pathway that we've studied quite extensively. I think that's JAK2 mediated, so a number of these pathways would be impacted. So I think there would be some sort of balance to be had, and we'd need to work out what was the right part of that balance to come out on, if you will. I think, but I think it's something we need to consider.
Thanks. So Chloe's got a hand up. Chloe, please.
I'm not interfering with anyone non-Exeter based. I don't want to ask, but as long as no one else has got a question, I'm asking this from the perspective of kind of beta cell replacement therapies, Mark, as a really interesting kind of overview and really nice to hear. But how do these sort of other cytokines, for example, some of the more aggressive cocktails of cytokines you'd see after beta cell or really islet transplantation, does, for example, a TNF alpha in combination with IL-1 beta have a synergistic effect with the interferons in terms of STAT1, or do they actually prevent that massive upregulation you see with interferon? So, in other words, how well does this translate to sort of transplantation therapies? That was a bit muddled. I hope that made sense.
So sort of asking about whether some of these other pro-inflammatory cytokines might contribute to the hyperexpression,
or the opposite. I mean, for example, I think I'm asking this question because I think, you know, if there are some other interesting pathways, for example, where I think TNF alpha prevents the upregulation of IDO1, for example. I may have that wrong, but I'm just wondering what we know about that and the interactions between the interferons and other.
I think the interaction of some of these pathways is fascinating, and it's not something we've specifically explored, but certainly would be worth having a think about, because there does seem to be a lot of interplay between these pathways to sort of generate outcomes. So the likelihood is there would be some effect there, but quite what level that effect is, I don't know. I haven't got a clue.
Okay, be really exciting to find out. If I see anything, I'll let you know as well. Thanks.
Thanks, Sarah.
So just really to kind of add on to Chloe's comment, I think this is something that Decio Azeric tried to look at relatively recently, and they did some studies. I can't remember all the details, but basically they had a look at whether IL-1 beta and TNF and interferon gamma and interferon alpha on their own could cause things like upregulation of class 1 and upregulation of other kind of hallmark molecules that we see upregulated in type 1 diabetes. And I think the kind of general message from recollection is that it's mainly the interferons, and that TNF alpha and IL-1 beta did not so much, but I don't know how much they did in combination there. So it certainly seems like the interferons were the stronger ones, for at least for the HLA component.
I actually remember this in a sugar science talk as well, Sarah. Thank you for reminding. I'll go back to it. Thank you very much.
I need to go back to that. I can't remember that one.
I think it's a fairly recent one, Mark, that's just come out.
I do have a Decio paper in my book here, which I haven't got around to reading, and I bet it's that one.
Yeah, I think it might well be.
Good, thanks, Chloe. Any other comments or questions? Can I throw one in about the elevation of total STAT1, Mark?
Yeah.
Do you think that is regulating gene expression in some other way than the phosphorylated version is? In other words, you showed in those plots the early phosphorylation, which is classically the activation mechanism, which then goes away after 24 to 48 hours, and yet the total level is massively elevated. Do you think that's controlling genes in a different way?
I think certainly there is evidence to say that unphosphorylated STAT1 can control gene expression. So by elevating total STAT1, it may well be that this is how this sort of elevation of HLA1 is being maintained through that process. Exactly how that works in terms of where things are binding, I'm not 100% sure on that, but that is certainly been described in the literature before, and that's one of the things that I was hoping to explore in a few grants that I put in that haven't quite been fully well, haven't been funded yet, but hopefully at some point we'll be able to explore that in some detail.
That's always referees never appreciating the significance.
Yeah, I also had a question about sort of any comments in terms of protective pathways, specifically PDL1, for example. With the JAK inhibitors, do they help to maintain those kind of sort of PDL1 protective pathways as well, or how does that fit in with combination therapies, you know, the mesenchymal stem cells alongside STAT1 inhibitors or something?
I think this is a great question. It's so similar to what Noel was saying earlier. I think we have to be really careful when using these, and maybe there are better ways of doing it, more subtle ways of doing this that aren't quite, because this does feel like a massive sledgehammer approach to some degree, because these JAK proteins are so ubiquitously expressed and they control so many different pathways that we potentially are going to be having all sorts of contrary effects. So yeah, understanding some of those, the effects, the less desirable effects potentially might be important to explore, as well as kind of looking at all the cool things that these JAK inhibitors can do.
Yeah, I guess I'm more excited from the perspective of combination therapies and what, you know, what else we can target alongside.
Yeah, I think these will be effective in that way, because I don't think, I mean, I think on its own, all this sort of approach will do is delay diabetes onset anyway. So I think if you are hitting it from other parts, you will be able to be much more effective. So in the context of maybe, you know, transplant and things like that, this might be a helpful way of reducing that or lengthening the time that the graft is viable for, those sorts of things. So I think this definitely probably will work in combination with things better than just on its own, perhaps.
Perfect, thanks.
Good. One final question from anyone? Please feel free. Sarah's got another one. Excellent.
Yeah, sorry, Mark. Just a little bit of a reflection on the kind of genetic predisposition to type 1 diabetes, and thinking about, because some of the JAKs, and particularly TYK2, comes up as a, you know, sort of a gene that's been associated with development of type 1 diabetes. Do you think that might give us some clues as to which JAKs are better to target?
Well, that's always the thing, isn't it? So it's the TYK2 thing it does sort of suggest that is something we should think about. And Desio had that paper recently where he was looking at the TYK2 inhibitor, which looked very beneficial. So I don't think we should be ruling any of those things out. I think the efficient approach, and the one that makes most sense, is to say, well, the JAK1 inhibitor hits both of those pathways, so it's probably the one that we should go with. But that's assuming that both of those pathways are equally important, and it's also assuming that both of the JAKs associated with those receptors, inhibiting them has an equal effect. And I don't think we really know that. So I think we certainly shouldn't exclude TYK2 inhibition. I think there are TYK2 inhibitors that are in development and could be used.
Yeah, and I guess another thing to think about there is we have to think about off-target effects there as well. So what we don't want to be doing is making people more susceptible to viral infections or reactivation of kind of latent viral infections. So that has to be considered. And I think Marlene Flodström-Tullberg has just published a paper that looks a little bit at the impact of some of the JAK or TYK inhibitors on susceptibility to viral infections in a gut cell line, I think. So might be worth having a look at that, because it might help you to tease a few extra things out. But I think it's literally just come out, so worth having a look.
Yeah, thank you.
Okay, good. Well, thanks a lot, Mark. So I'll reserve stories about the 20 years since you joined the lab for another day, not least one in Tampere, but we'll talk about those again. Thanks, that was a really good talk. So I'll hand back to Anna and Sarah. Thanks.
Thanks, Noel and Mark. That was such a clear talk. Thank you. As Sarah alluded at the beginning, as some of you were joining, we're hoping to have another meeting and talk on the 3rd of June at 1 p.m. That's a Monday. But as yet, we've not got a presenter. So if you have some recent data that you'd love to share with people, or a story you've been working on for a while that you'd like to share, then do let us know, and we'll see if we can drum up a speaker for that session as well. Sarah, was there anything else you wanted to add?
I don't think so, other than to kind of highlight, we'll put some of the updates of speakers, and I suspect Anna and I will come chasing a few people, and I've got my eyes on someone in particular, so sorry to that person in advance. I might be calling on you shortly, but we'll put the updates in the link on the T1D Egg website, and the link for that is in the email that Anna sent through with the invitation to this meeting. So please keep an eye out for the updates there, and we'll also send an update around by email. But look forward to hopefully seeing you all next month.